you so much for the invitation to be here. It's great to be addressing such a diverse audience at this conference. What I'm going to talk about today is an area of biocatalysis that's really intrigued chemists for several decades, and that's the biological activation of hydrogen. This is really widespread. Is that still working okay? Yeah. It's really widespread in the microbial world, in all sorts of environments. And in fact, as we're digesting our lunch, we will have microbes in our gut that are producing hydrogen from fermentation and others that are using that hydrogen for as an energy source. And so I'm going to talk to you today about some of the work we've done to probe the mechanism of hydrogenases, and that is coupling electrochemistry, structural studies, and spectroscopy. And we show that bringing together all of those three techniques has been really powerful in probing the mechanism. I'm also at the end just going to touch on a little bit of the applied work that we've done to show that enzymes can actually make it into the commercial world as catalysts that have application in chemical synthesis. So the enzymes responsible for biological hydrogen activation have either an iron or a nickel-ion catalytic site, and we're particularly interested in the nickel-ion enzymes because they tend to be more robust towards oxygen and hence more applicable for biotechnology purposes. And the catalytic site of these enzymes is built from a nickel and iron in this bimetallic active site that's deeply buried within the protein. And you'll notice that it has biologically unusual carbon monoxide and cyanide ligands that we rarely find in biology, but they are critical here to tuning the active site to turn it into a good catalyst for hydrogen activation. What's really impressed chemists is the fact that nature has found a way to use nickel and iron for activation of hydrogen at turnover frequencies that rival platinum. Of course, the enzyme has only a single active site within the protein structure, and you can think of this as a nanoparticle catalyst. The enzyme has a diameter of about 5 nanometers, so it's really a nanoparticle catalyst, but with just one active site finely tuned and buried inside this protein framework. Of course, a platinum nanoparticle of a similar size would have many active sites on the surface, so it doesn't do so well on a sort of activity per volume basis. As well as having this bimetallic active site, the enzyme has a chain of iron-sulfur clusters that allow electrons to move very rapidly between the buried catalytic site and the surface of the protein. And it also has channels that allow rapid access of hydrogen and protons in and out of the active site. And these enzymes often function as reversible catalysts for hydrogen cycling, able to oxidize hydrogen and reduce protons to make hydrogen. And I've added on various slides a reminder to myself to acknowledge the people who've contributed to this work. And we're really indebted to some early microbiological work from Frank Sargent, which provided the strains for expression of the hydrogenases from E. coli, which have turned out to be really good workhorses for probing the mechanism of hydrogen activation in hydrogenases. And I've also included here an acknowledgement to Non Evans, who is a postdoc in my group who did a lot of the strain development. So the mechanism for nickel-ion hydrogenases, as we know it, starts from this nickel-2, iron-2 intermediate with a well, I'll come back to this, but likely a vacant coordination site here between the metals. And hydrogen is cleaved heterolytically, unlike on a platinum surface, to give a metal-bound hydride here, the bridging hydride, and a proton on a nearby base. And that is still under contention, the site of that proton. And there, I guess, a lot of work has suggested that it could be one of the cysteine sulfurs coordinating to the nickel that accepts that proton, whereas an alternative theory has been put forward that an arginine nitrogen group that lies above the active site could be the site for that. I'll come back in a little while about some of the ways that we might be able to approach that question. The remaining steps in the catalytic cycle involve a series of proton-coupled electron transfers where in this first step here, the proton that was released from hydrogen cleavage moves out through a series of amino acids or ordered water molecules in the protein and leaves the active site region, and an electron is also transferred out through the chain of iron sulfur clusters, leaving the nickel oxidized up to nickel 3. There is then a tautomerization step where the hydride leaves as a proton onto again a nearby base, and the nickel is reduced down to nickel 1. The base for this step is likely to be the carboxylate side chain of a glutamate residue, and I'll show you later how we have direct evidence now for that glutamate being involved in proton transfer. And then finally, another proton-coupled electron transfer step removes the proton off the active site and reoxidizes the nickel back up to the nickel 2 level, ready to start the catalytic cycle again. And our insight into this catalytic cycle has been built up from a mixture of EPR and infrared spectroscopic studies in particular, as well as X-ray diffraction structures and computational work. And as you'll well recognize from other work in catalysis, the CO and CN ligands at the active site of hydrogenase provide ideal probes in the infrared for looking at the active site and are very sensitive to any changes in electron density on this site, meaning that we can track every single intermediate in catalysis via the CO vibrational band position. Is the microphone sounding okay? It's a bit boomy from here, but you can hear okay, hopefully. Great. So we can also think about sort of moving through or navigating through these different states under electrochemical control, because each of these steps in the catalytic cycle is simply transfer of protons and electrons. And so this is work that's been done on a number of hydrogenase enzymes in solution where an electrochemical potential has been applied, and you can use that to navigate from the most oxidized down to the most reduced state of the active site. We've been able to do that under catalytic control on an electrode surface, handling the hydrogenase as an electrocatalyst. And in that way, we can use electrochemistry as a very valuable tool to control the state of the enzyme during catalysis and during spectroscopic measurements. There is also a further oxidized state of the active site, which is not within the catalytic cycle, but can be generated under conditions of oxidative stress in physiologically relevant conditions, and generated by oxidation or by reaction with oxygen in the enzyme when it's isolated from the cells. So as I mentioned, each of these states of the active site has a characteristic CO vibrational band position, and we can then use those to be able to determine which active site states we have present under any particular set of conditions. And you'll notice here that while some of these states have a unique CO band position at the nickel L redox level and the nickel R redox level, we've actually got a mixture of different substates, and we think that those differ in the position of the proton close to the active site, and that CO vibrational band is so sensitive to changes in electron density that it can sense a proton moving in the vicinity of the active site. Now, the experiments I'm going to talk about now, and for the next little part of this talk, come from electrochemical control over crystals of hydrogenase. And we started these experiments as one of those sort of, why don't we just try this and see what happens kind of experiments that actually worked absolutely brilliantly the first time, one of those sort of strokes of research luck that you get every now and then. So we were doing some different experiments on the infrared microspectroscopy beamline at the UK synchrotron diamond light source. And while we were there, we had some crystals of hydrogenase that Stephen Carr, who's a senior research fellow and protein crystallographer within my group, Stephen Carr had for X-ray diffraction studies on hydrogenase. And a brilliant postdoc in my group at the time, Philip Ash, who's now a lecturer at the University of Leicester in the UK, Philip said, why don't we try putting some of these crystals of hydrogenase onto an electrode and applying a potential and seeing whether we can actually manipulate the crystals electrochemically. And it could have gone wrong for lots of reasons, but it actually worked absolutely brilliantly. And these are not quite the first set of spectra we recorded, but they look very similar to the very first spectra we recorded in those sorts of experiments. And so what you see here is that we're looking at the CO vibrational band, again from the active site. The cyanide bands are a bit further up here. The CO vibrational bands, as we step the potential for an electrode in contact with a single crystal of hydrogenase, and we're using the infrared microscope to focus the infrared on a little spot on that crystal. And so this is the crystal, a needle-like crystal lying on the electrode surface here, and the little black square zoomed in a bit more here is the area that we're sampling with the infrared, which is about 10 by 10 micrometers. And so we're able to start from the most oxidized state of the active site, this oxidized inactive state known as nickel B, and then as we step the potential down, we go through all of those different states relevant to the catalytic cycle that we've seen in solution or on an electrode surface, right down to the reduced nickel R states here. And that was really exciting because it suggested that it might be possible to dial up crystals in really specific redox states. And that's a real challenge if you're trying to do structures of protein crystals, because it's very easy to end up with a really complicated mixture of states where you just don't know what you have present, and of course you then see an average of all of those species in the electron density map. So if you just add a reductant or add hydrogen, you're likely to hit some potential where you've got a real mixture of states, whereas with the electrode potential we can dial up very specific states. Now I'll just step back for a moment to show how that works. We've built this cell in-house, and we have a mirror-polished carbon electrode, and the crystal sits on that electrode, and as we apply a potential at the electrode, the crystal then responds to that. We handle the crystal in its crystallization buffer, so the liquor that the crystal was formed within, and so it sits on the electrode in a little pool of that crystallization buffer in contact with a counter and reference electrode. And we add to the crystallization buffer a series of redox mediators which give us access to the whole potential range relevant to the experiment that we'd like to carry out. And so this is the electrochemistry of the series of mediator molecules that we've used here. In this case, they're all organic mediators, but to get down to lower potentials, we also sometimes use europium ligand complexes, which give us a nice range of tunable potentials in the more negative range. And the mediators are able to diffuse into water-filled channels within the protein structure. So Steve prepared this image showing the unit cell for hydrogenase with the hydrogenase molecule and the water-filled channels which contact the sort of edge of the crystal with the surface of the protein where the iron sulfur clusters allow electrons to exchange, so that you can equilibrate right through to the active site very rapidly within seconds to minutes of applying a potential at the electrode surface. The crystal doesn't have to be actually touching the electrode, it's just sitting in that solution in contact with the electrode. We did find we got some redox control if we didn't have the mediators there, but it was very slow, so they helped to speed up that redox transition. And so we can record this sort of set of spectra as we apply potentials at the electrode, and we can think of this as a route map that allows us to map out exactly what potential we have to dial up to get any state we want to in the hydrogenase active site within the crystals, and we're getting complete conversion of all of the hydrogenase active sites within the crystal into the states that are represented here in the spectra. And they're lovely intense spectra for a protein molecule, because within the crystal we have an effective concentration of about 8 millimolar protein, which is much more than we could ever get in solution. And so that gives us these very nice looking spectra. So what I'm going to show you now is a series of as yet unpublished structures for hydrogenase in a whole range of redox states, where we've used this route map as our way of dialing up the state we need to get to structures of all the different states of the hydrogenase active site. And just to sort of follow on the route map kind of idea, what we've done here is to plot out the intensities of the bands, giving us a measure of the absorbance, giving us a measure of the concentration of each species that we have at each potential. So this is potential along the x-axis here, and the concentration of different species shown in colours that map onto the colours of this catalytic cycle image over here. And what really struck me was that this top one is in crystals, the middle one is with the enzyme in solution, and this bottom one is with the enzyme adsorbed on an electrode surface. And the speciation curves look almost identical between all of those. And that shows us that we can really think about this crystal environment as being very relevant to the behavior of the enzyme in solution, or indeed on an electrode surface, if you like. But it shows us that we're really getting dynamic behavior of the protein in the crystal, which allows us to sort of navigate between all of these states that we observe in solution and that are relevant to catalysis. That may be somewhat unique to hydrogenases because, of course, they have the smallest possible substrates of protons and hydrogen, so you don't need a major structural change to be able to bind substrate and transform it. In fact, you really don't want major structural changes if you're trying to do very fast redox catalysis, and I'll come back to that later. So we start with the nickel Si state at the beginning of the catalytic cycle. And what we've done here is used our electrochemical approach to dial up conditions that give us a very pure nickel Si state here. And we then extract crystals from that poised solution and determine the X-ray diffraction structure of those. There was a lot of reasons why this might not have worked. We were unsure whether the crystals would maintain their diffraction quality after they'd been subject to electrochemical control. And so we were really delighted when we got a 1.37 angstrom resolution structure of this nickel Si state, where we'd lost no diffraction quality at all following the electrochemical control. And this, the only other structures of this state that exist are one from photolysis of a CO inhibited form of the hydrogenase, or one from a very unusual hydrogenase where we know very little about the other states of that enzyme. So this was quite exciting to get this nickel Si structure. The nickel displays the seesaw geometry, which has been predicted from computational work to be optimal for hydrogen splitting. The active site is generally thought to be empty in nickel Si, so it's normally represented like this ChemDraw image over here, but actually we do see electron density between the metals here, which would be consistent with partial occupancy of a water molecule, and that would tie in with what we're seeing in the spectra here, where there's an extra band in the cyanide region. We should only have 2 cyanide bands, and probably what we're seeing is the overlap there of of a water-bound and an empty active site there, where they're similar enough in electron density that the CO bands are overlapping there. So we then move on, well, I guess to confirm, that was really at the nickel Si redox level. We reacted the crystals with carbon monoxide when we poised them electrochemically at that level, and we now have a very obvious extra peak in the spectra, which is consistent with the exogenous CO molecule bound to nickel that's well known, this CO inhibited state. And in the structure, we now have electron density consistent with the CO inhibitor bound to the nickel in exactly the position that would be expected and has been observed in other structures for CO-bound hydrogenase. This is all for an enzyme called hydrogenase 2 from E. coli, and I'm going to make use of 2 different E. coli enzymes in this mechanistic study because they have slightly different stabilities for different states in the active site, which allows us to get at different catalytic intermediates by choosing either HYDE2 or HYDE1. Interestingly, HYDE1 has been thought to be almost completely insensitive to CO, like many other very oxygen-tolerant hydrogenases. But in fact, when we dial up the nickel Si state, as a starting state and then react it with CO, we were also able to get a very similar structure for HYDE1 completely inhibited by CO. So this is also quite a useful gateway approach to electrochemically dialing up a specific redox state, which then binds to an inhibitor. And you can imagine a lot of ways that that could be used in studying particular inhibitor or ligand-bound states of metalloenzymes. We then move on to the reduced nickel R states. Now in the CO region, we do have a mixture of 2 states, and we think these are these 2 protonation substates that we label nickel R2 and nickel R3. And we then, when we record the structure of this, we see electron density now between the metals, which would be consistent with a hydride that we expect in the nickel R states. And this superimposes beautifully with the hydride position in a super high resolution structure of nickel R, reported by Agata's group in 2015, where that was likely a mixture of all the different nickel R states. So that places the hydride within the metals, the bridging position as expected there. Obviously, X-ray diffraction is not the ideal method for trying to look at the positions of hydrogen atoms, and I'll come back later to how we might be able to actually pinpoint some of these. We'd really like to know where this proton is in these two states labelled nickel R2 and nickel R3, and I'll come back to that briefly later on. We then move around the catalytic cycle to the nickel C state, where we have now gone to a nickel 3 state, so the nickel's oxidized, and we maintain the hydride bridge as we expect. And interestingly, the metal-metal distance has not changed at all in these structures, despite the loss of an electron from the active site. And I think that really shows the impact of the redox buffering of the combination of pi acceptor sigma donor ligands for the CO and cyanide on the active site, which allows you to be able to add a whole electron without actually moving the metal distances at all. And so this is an incredibly rigid catalytic site. Now, before I move around to the next bit in the catalytic cycle to show you the structure at the nickel L level, I'm just going back a step to some work that we did a few years ago. Philip Ash, while he was still in my group, worked with me on this. And we analysed all of the infrared data that was available for hydrogenases from different organisms to look at the positions of the bands for these nickel L states, where there's often multiple CO vibrational bands suggesting different protonation states at that nickel L level. And I won't go through all of the analysis in detail, but in brief, these nickel L bands fall into 3 groups, which are consistent with what's predicted from modelling studies of the movement of a proton starting on the active site, a proton near the active site, and a proton far away from the active site where it no longer influences the CO vibrational band position. So this is the sort of most electron-rich active site with the lowest wave number CO band, if you like, because the proton has now moved far away. So that gives us a marker for which of the nickel L states we have present, whether we have the proton on or very close to the active site, a bit further off the active site, or far away. So the first thing we've done here is to photolyze crystals in the nickel C state. And it's been known for a very long time that the nickel C state is light sensitive, and when you shine light on it at cryogenic temperatures, you can convert it into nickel L. And in fact, nickel L was originally thought to be an artifact, and the L comes from L for light. It was thought to be an artifact that you only generated when you shone light on nickel C. Since then, we've shown through catalytic studies under operando conditions that nickel L is actually an intermediate in the catalytic cycle of hydrogenases. And we think that this state that you get from photolysis is the first proton position. So the hydride has just moved, that hydrogen atom has moved off as a proton onto a residue on the active site or very close to the active site. And we don't, obviously, from X-ray diffraction, have direct evidence of where that proton goes, but we do see an increased mobility of the cysteine residue 546, which is one of the cysteines coordinating the nickel that would be consistent with that cysteine having taken up the proton in that first step. Interestingly, I've also included this glutamate carboxylate side chain here so that I could point out to you that we see no change of the position of that side chain in this nickel L1 structure relative to what we saw in the nickel C structure, and that's going to become important in a moment. So that is consistent, the position of that glutamate side chain is consistent in every single one of the intermediate states of hydrogenase that we've seen so far. So this is where it gets a bit more interesting, because up to here we were sort of thinking this is all pretty boring. We've done all these structures and put all this effort into electrochemically controlled structures, and we've actually seen almost no movement at all. There is no movement of the metals. there is no movement of the primary or secondary coordination sphere. There's no movement further out. This is all sort of a bit boring after all that effort. And then we saw something interesting. In this case, we've now turned to hydrogenase 1 from E. coli, and this enzyme has stable nickel-L states which are present at room temperature and in the dark, so we don't need to illuminate to get to these states. And we've got a reasonably high proportion of nickel-L in the crystals. We always have a bit of some other states there as well, but we've got quite a high proportion of nickel-L here, enabling us to get a structure at reasonably good resolution of nickel-L. And this is from the infrared spectra. We can see that this is a mixture of nickel-L2 with a little bit of nickel-L3 present. So we've now moved that proton a little bit further away from the active site. And the really important point here is that this glutamate side chain has now undergone a little flick to bring its position close enough for hydrogen bonding or tunneling of a proton directly from that cysteine residue to the glutamate. And that sort of fills in a gap in understanding of the hydrogenase catalytic cycle, where previous work had shown that tunneling was likely in that proton transfer step. A lot of work, mutagenesis, computational studies had suggested that the glutamate was important in the catalytic cycle, but it was unclear how it could possibly be involved when it was too far away to engage in proton hydrogen bonding or proton tunneling. The distance in the other structures is all around 3 angstroms or just over 3 angstroms. And here we bring it just close enough at 2.7 angstroms to be within the distance that people like Judith Klinman have modeled or measured from studies of mobility within proteins is critical to allow proton tunneling without needing major structural change in a protein. So I think this gives us really direct evidence that that glutamate residue can be involved in proton transfer in hydrogenase. And it shows us that we've got this active site that is absolutely rigid, and what nature has done is found the most subtle movement possible in order to allow proton transfer, which does need a little bit of mobility within a protein. So I think this is an enzyme that's tuned for very fast, low reorganization barrier, according to Marcus theory, electron transfer, but very subtle movement to allow proton transfer. So that was quite an exciting finding there. And I just show these structures overlaid here. This is the structure of the nickel C state overlaid on the nickel L2 state. And you can see that the way that that glutamate flick has brought it from being 3.6 angstroms to 2.7 angstroms from the active site. And it's absolutely conserved in this position in all of the other states, and just in this nickel L2 state it comes within hydrogen bonding distance here. So to summarize this part of it, what we've really got here is a series of structures that we can now paste onto this cycle that otherwise is just a series of ChemDraw images pasted together from lots of different pieces of work. We now have structures that show us what's happening here. They show very minimal movement, but this really key flick of the glutamate, which is key to fast proton transfer within this very finely tuned catalytic cycle. Just very briefly, we have now started doing some room temperature structures using the XFEL source at LCLS, working with Jan Kern and his team, and in a similar way have been able to dial up electrochemically redox states in microcrystal slurries. So this is the series of spectra on different slurries of microcrystals that we've tuned at different redox states, ready to record XFEL structures, and we've been able to get good resolution XFEL structures of all of those. So these are now room temperature sort of ground state structures, which provide the beginning for being able to then do some time resolved studies, either by photolysis or triggering of reactions in other ways, to be able to look at reactivity using the X-ray free electron laser as a way of getting time resolved structural information at room temperature. Interestingly, the structures map on very well. They also confirm that our other structures recorded on the synchrotron are damage-free because the XFEL records the data so quickly before the crystals have time to be subjected to X-ray damage, so that this gives us confirmation that the structures we were looking at before are all avoiding any photon damage that can be a real problem in metalloprotein structures. So this is sort of the start of some time-resolved work that we're moving towards now on the XFEL. I'm going to skip over that one. Of course, X-ray diffraction is not the best way to be looking for protons in structures, and we're never going to see that. And so the other thing we've been doing at the other end of the crystal size dimension is growing very large crystals that we can use for neutron diffraction. And we've been able to collect at the nickel C redox state the first diffraction data on these large crystals at the neutron source at ANSTO in Australia, and we have further beam time coming up at Oak Ridge later this year. So we're hopeful of being able to begin a series of neutron structures. This is a huge piece of work because we need enormous crystals, and it takes about 120 litres of E. coli growth to get enough protein for the crystals for neutron diffraction. But we're hopeful that this will then start to tell us about where these protons and hydrides are definitively within these structures. In the last few minutes, I'm just going to show you a little bit about what we can do with all of this and why we might care about structures and mechanism of hydrogenases. And so this is quite a diversion because this moves into the biotechnology area, where hydrogenases offer us the possibility of cleaning up biocatalysis using hydrogen as a clean reductant, just as it is in hydrogenation reactions in heterogeneous catalysis. And so what we're really trying to do here is to turn biocatalysis into heterogeneous hydrogenations. And we've done this in a number of different ways, but the reaction I'm going to start with here is the reduction of nitro compounds to amines. This is a very key process in pharmaceutical synthesis. Many, many pharmaceuticals are made by a palladium-catalyzed nitro reduction, and hence we often end up with palladium left behind in paracetamol from the palladium-catalyzed nitro reduction, for example. And so there is a need for cleaner, more selective ways of doing those reactions. Nitro reductions can happen on a carbon surface, and a lot of the work we do is with enzymes immobilized onto a carbon support. And so we hypothesized that the reduction potential for the nitro group on aromatic compounds is compatible with the hydrogen potential, and that we should be able to set up a catalyst that involves hydrogenase on carbon, where the carbon support is able to reduce the nitro group. And we do these reactions in much the same way organic chemists might run hydrogenation reactions. This is a little octo-reactor of parallel vessels, each with this black powder enzyme on carbon catalyst under a hydrogen atmosphere, just run in a small aqueous solution with a little bit of organic phase to help or organic solvent to help solubilize the organic compounds. And so we've shown that this works on a wide range of different nitro compounds, and I'll just pull out a couple of examples of why we've picked certain functional groups here. We've done this with sulfur containing nitro compounds because those often poison a palladium catalyst and hence cause problems using conventional metal catalysts. We've done this on other unsaturated functional groups on the nitro group, but with molecules that have other unsaturated functional groups like alkynes, ketones, carboxylic acids, alkenes, and all of those selectively reduce the nitro compound without touching the other functional groups there. We've done this on dinitro compounds and been able to reduce both of those nitro groups, and we've done this on halogenated compounds where palladium will often dehalogenate, but in this case we have full retention of the halide co-substituent there. And we've done this on a sort of slightly scaled up to gram scale synthesis to show that you can make a pharmaceutical molecule using this and on sort of a scalable quantity. And this catalyst system is now licensed to the spin-out company Hydrogen and is now being commercialized for application in fine chemical manufacturing. This is very readily translatable to flow because the enzyme's immobilized on a carbon support, and so this is a flow column incorporated into the commercial hydrogenation reactor, which produces hydrogen from electrolysis of water, and this is the hydrogenation of nitrophenol, which is to generate the aminophenol, which is relevant to paracetamol synthesis in continuous flow. And what we see, this was a very, very preliminary experiment without any optimization, and we straight away got over 70% conversion running for 4 days there and giving us over 1.5 million turnovers for the hydrogenase during that time. So this is looking really promising. Now, just very finally, going back to the sort of electrochemical way of thinking about this, the hydrogenase that we've used for this initial work was hydrogenase 1 from E. coli, one of the enzymes we've done lots of structural work on, but it has a small overpotential for hydrogen oxidation, meaning that it's losing a bit of that driving force from hydrogen. And we plotted the onset potentials for reduction of a wide range of nitro compounds, and all of these aromatic nitro compounds fall above the onset potential for HYDE1, showing that HYDE1 should be compatible with being able to reduce those, and indeed we see hydrogenation of the nitro groups for all these compounds. But when we want to look at aliphatic nitro compounds, like nitrohexane here, we need to turn to hydrogenase 2 from E. coli, which is not quite so robust, but does work right at that thermodynamic potential as a reversible catalyst. And so we hypothesized that it should be possible to put HYDE2 in place of HYDE1 on this sort of system and be able to extend this to aliphatic nitro compounds. And indeed, here are some NMR spectra showing that we are successfully hydrogenating the nitrohexane using HYDE2 on carbon catalyst. So it's a nice example of how the electrochemistry of the enzymes allows you to design a catalyst and tune what you're doing. So with that, I'm going to thank all of the people who've been involved in this work. My group here, in particular, Philip Ash, who's now left the group, and Non Evans, who've now left the group, who've done a lot of work on the mechanism side of things, and Sophie Kendall-Price, who's now a postdoc in York, and Wangji Li, who really got us started on the crystal, towards the crystal structures, and more recently, Kin-Long Wong, who is a DPhil student who's just coming to the end, who's done a lot of the, I guess, recorded most of the structures together with Steve Carr that I've shown today. And on the biotech side, we've had some great collaborations with Johnson Matthey and with Hydrogen, who've worked with us on that. So thank you very much.